Well, I wanted to tell you about research, and specifically research as an undergraduate experience and as a component of your education. I'll give you some opening remarks, talk about general principles of research, and then talk about some specific examples, including some Creighton undergraduates and what the impact of an undergraduate research experience has done to their career paths. And you're lucky at Creighton because we have such a complex campus that it doesn't make any difference if you're interested in biomedical research, political science research, business research. We have it all because of the complexity of this dense campus. And a lot of times you can go from political sciences to biochemistry to biology without even going outside. You can just move around so quickly. So first, let's just look at the general principle. What is research? And as I've indicated here, and you probably know, research is really a process of investigating hypotheses, or maybe just suggesting a new interpretation of data or text that already exists. It usually means posing a new question and new questions for future research to explore. And the driving force for research is curiosity. So if you're curious, usually you can find some discipline and research in that discipline in which you can immerse yourself, be engaged, and find fulfillment. So you want to try to ask a question nobody's asked before, and if you're lucky, you might be able to answer that question, but for sure you can ask the question. You want to do the necessary work to find out the answer, and you want to also to be able to communicate the knowledge that you've acquired to a larger audience. One of my postdoctoral mentor told me once that the best experiment in the world and the results of that experiment don't do any good if you just leave them on your desk. You have to communicate the results to the world.
It's a sunny day in San Francisco. We set up a table by the water and asked people this question. If it's the year 1960 and you had $10 to give to science, would you spend it on A, developing an affordable treatment for diabetes, or B, figuring out how bacteria protect themselves? The majority of people we surveyed wanted to spend their money on a diabetes treatment, but what really happened 50 years ago? The government decided to spend money on figuring out how bacteria protect themselves against viruses. The National Science Foundation, the NSF, and the National Institutes of Health, NIH, are two government agencies that allocate tax dollars to scientists. And they decided to fund groups of researchers who thought that studying how microbes protect themselves was an interesting question. At the time, there was no way of knowing how this basic research could affect human health. They were simply curious about understanding something about bacteria biology. So what did the scientists find? Normally, when a virus enters a bacteria, it adds its own DNA to that of the bacteria. In doing so, the virus takes over the bacteria and uses it to make more viruses. Scientists found that bacteria protect themselves from viruses by selectively cutting the viral DNA. This basic finding led to a scientific revolution. Scientists learned how to cut specific chunks of DNA, and they could use this technique to move genes around. They could even move human DNA into bacteria. And because bacteria are very good at making more of themselves, scientists can use them to make large quantities of human proteins from the human gene that they introduced. So why was this technique useful for modern medicine? Well, one breakthrough was the efficient production of human insulin by introducing the human insulin gene into bacteria. This was a very significant advancement in diabetes treatment. Before using bacteria to produce large quantities of human insulin, insulin was expensive, unsafe, and less effective because it was harvested from fetal cows. In fact, the first person to get an injection of cow insulin suffered from a severe allergic reaction. Because of a long line of federally funded basic research, human insulin is now an affordable treatment for diabetes. By asking how bacteria protect themselves, the scientists discovered that bacteria can cut DNA. This discovery was developed into a basic technique used in thousands of labs around the world to create new knowledge and to ask new questions that have led to all kinds of therapies and technologies, including insulin, DNA fingerprinting and biofuels, the human genome, growth hormones, cancer drugs, stroke medication, vaccines, HIV meds, antibiotics, and many, many more. Given enough time and funding, who knows what is to come. Many other health benefits and current discoveries would have been nearly impossible without federal funding for all kinds of basic research. Basic research is an investment in our future. It broadens our understanding of the world we live in, and the potential benefits are limitless. Unfortunately, science funding in the US has plateaued, decreasing purchasing power and significantly hurting our competitiveness and innovation. You may not know it, but you and your tax dollars are part of the discovery process. Thanks to you, scientists discovered that bacteria can cut DNA. And thanks to you, insulin, other drugs and diagnostic tools have been developed and are helping patients.
So thank you for supporting basic research.
We are the scientists who depend on you. And we are excited for the future that lies ahead.
I love that video. So sometimes it's not possible to predict what the impact of basic research is going to be. So this is a case of biomedical basic research and all of the discoveries that ensued after that. But again, not all research has to be basic. There's clinical research, there's political science research, there's Romance languages research, but this is one example of the impact, tremendous impact of biomedical research. This little clip was produced by those students or graduate students at the University of California in San Francisco just last year. So let's get to undergraduate research. So this is an opportunity to engage in a sustained and progressive faculty-mentored project, research project. And so it's an opportunity to develop kind of a long-term relationship with a faculty member at Creighton, again, irrespective of the type of discipline that you like and want to engage in. In undergraduate research, it's an example of what's called a high-impact practice, a high-impact educational practice. And these are practices that require considerable amount of time and effort, but they provide learning opportunities outside of the classroom, certainly produce meaningful interactions between faculty and students, and encourage investigation and critical thinking, and faculty then can give you meaningful feedback and as well as mentoring. So undergraduates can begin to complete a research experience even in their first year of attendance at this institution. And that can last through the time that you graduate. Dr. Sukup does a wonderful job of organizing this now through CURIS, the Center for Undergraduate Research and Scholarship. So pursuing research can be a challenging endeavor, from deciding on a topic to establishing the appropriate protocol and methodology to use, from analyzing data and then planning further research on the topic. And again, as I keep telling you, it's not related to just one discipline or department at Creighton. You can pursue research across the disciplines, the sciences, the humanities, social sciences, all the way to the clinics because of the complexity of our campus. So I want to give you a few examples of the impact undergraduate research has had on people, starting with myself. So I don't like to ask students to do things that I don't do. I don't like to ask faculty members, for that matter, to do things that I don't do. So I try to lead by example. So when I was an undergraduate at the University of North Texas, I worked in Dr. Robert Gracie's lab. He was a young faculty at the University of North Texas at the time, biochemist, trained in California, went on to become a professor of biology and chemistry, and he served as chair of a biochemistry department, and then as the dean of research. And he's at the University of Texas at San Antonio now. So I worked in Dr. Gracie's lab as an undergraduate with my junior and senior years, and his laboratory had federal funding, like the graduate students in the little video clip talked about. And his federal funding was to study an enzyme, triose phosphate isomerase. And the rationale of a section of his grants was that this enzyme deficiency caused a severe enzyme deficit that resulted in either retardation or death at an early age in humans. So in his lab as an undergraduate research assistant, I learned to prepare buffers, to load columns, and because this was in the 1970s, all of our literature research, I had to read the abstract of the article that he was interested in and then summarize it on a 3x5 card and write out the full number of authors and the title of the paper, because we didn't have personal computers to store the bibliometric data electronically. But this experience, it really changed my mind. Before that, I was thinking about either applying to medical school or maybe being a school teacher. And because of this experience, I decided that I would definitely go on to graduate school, and that's what I did. So we moved from the 70s to the 1980s, and I was a young faculty member, and I had in a college of pharmacy, and a pharmacy student wanted to work in my lab as a undergraduate research assistant. He was an undergraduate. His name is Mark Lead. He's currently the assistant dean for scholarship at Oregon State University. So he had a publication as an undergraduate in my lab. He went on after becoming a pharmacist. He worked for a couple of years as a pharmacist, and then he went on to graduate school and received a Ph.D. in pharmacology. So his undergraduate research experience sort of terminated his career as a pharmacist, and he went on to get a Ph.D. in pharmacology. He then moved to France to do a postdoc in Pierre Chambon's lab in France, and he published a paper in 1992 talking about retinoic acid receptors, a receptor for endogenous hormone. And there's more than one of these retinoic acid receptors, and really what he discovered is that the two of them existed bound together as a dimer. It's a very important paper. And the next year, it was that paper that he published was characterized as the hottest research paper in all of life sciences because of the number of citations it received. And I checked last night as I was putting these slides together in the Web of Science, and as of last night, that paper has been cited 1,177 times. That's pretty incredible, very important paper. And I like to feel like I get a little credit for that since he worked in my lab as an undergraduate. Let's move to this century. 2003, I had an undergraduate working in my lab at the University of Georgia. His name is Chris Hale. He's from a small town in South Georgia. He worked in our group for 2.5 years, did all kinds of experiments, including this primary cell culture, utilized sterile technique, did some biochemical studies. And he was he majored in cellular biology and Romance languages, and he was also the director of the University of Georgia breakdance team. So he had a lot of different interests as an undergraduate student. He took the MCAT exam in 2003, and I kept on bugging him to say, Chris, how did you do on the MCAT? And he would never tell me. Then finally, in a very self-effacing way, kind of put his head down. I said, well, didn't you do okay? And he said, well, I got a 41. Well, that, you know, their percentile. So that year, a 41 was the 99.9th percentile. So he did pretty well. And he attributed part of his success, certainly not all of his success, to learning the nomenclature of science, learning the nomenclature of biomedical research and medicine really helped. Wasn't the only thing he did to prepare for the MCAT, but it helped him. He made several presentations research presentations as an undergraduate. He developed a project where he studied the effect of cigarette smoke exposure on nicotinic receptors in the brains of rat. Those are slides directly from one of Chris's poster. Today, Chris is a third-year dermatology resident in New York City and doing very well. But it started as an undergraduate researcher. Let's come to Creighton and the undergraduates at Creighton University. One undergraduate I want to tell you about is Mike Rukas. Mike is currently an assistant professor in the Department of Anesthesiology, Anatomy, and Neurobiology at Washington University in St. Louis. He got his BS degree from Creighton in 1999. He stayed on at Creighton and did a Ph.D. in pharmacology in the School of Medicine and graduated in 2004. As an undergraduate, Mike worked in Dr. Margaret Schofield's lab in the Department of Pharmacology. He worked there for a couple of years. As a graduate student at Creighton, he worked in Dr. Peter Abel's lab. Peter is still on the faculty here. Last year, Mike, he then went on to do a postdoc at the University of Washington after he left Creighton. And then for the last year and a half now, he's been on the faculty at WashU. He's received two very prestigious awards recently, federal grant awards, the NIH Pathway for Independence Award, and a very prestigious, extremely prestigious NIH Director's Transformative Research Award. Only about 10 of those are given out a year, really at the discretion of the director of NIH. So there's, you have to be really, really good to do this, and as a Creighton grad, Mike is doing very well. His laboratory, along with collaborators at the University of Illinois, some engineers from the University of Illinois, have developed tiny devices that contain light-emitting diodes so small that the size of individual neurons, and these devices can activate brain cells optogenetically with light. His team reported those findings in the journal Science in 2013. He's been on national webinars with some of the stars in the field, and himself has really become the equivalent of a rock star in science, and a fairly recent Creighton grad. Even more recently, in 2008, a Bachelor of Science grad from chemistry, got a BS in chemistry, is Annie Wong. Annie Wong went to high school in Lincoln, so she's a Nebraska native. She, too, stayed on at Creighton after her finishing her undergraduate degree and obtained a Ph.D. in pharmacology. At Creighton, she worked as an undergraduate in the chemistry department with Jim Fletcher. She had a couple of projects that I've listed there with Dr. Fletcher in the chemistry department, and really piqued her interest in research, and that's why she stayed on to do a Ph.D. The list of Annie's awards as a graduate student is really incredible. She won second place, the Best Abstract competition at a very large national meeting, competing with graduate students from Vanderbilt and Johns Hopkins and UCSF. She won the top, she won a travel award at a national symposium on prostate cancer, and she won the top award at that same symposium that she had received the travel award from. She received a couple of travel awards to go to additional meetings. The ASPET stands for the American Society for Pharmacology and Experimental Therapeutics. And she was, in 2013 at that meeting in Boston, and she again had the runner-up poster. Given all of her accolades via national competitions, she was awarded the Creighton University Academic Achievement Award as an outstanding graduate student in 2013. Her PhD as a graduate student at Creighton was in the cancer field. That's the title of her dissertation. And Annie is now a postdoctoral fellow at Stanford in the Department of Urology and doing extremely well. She's doing so well that she was thinking about doing a postdoc at another laboratory recently, and her current major professor and mentor begged her to stay on, so she's going to stay on at Stanford. So there's a lot of opportunities. Now, the examples that I've given you have a biomedical flavor to them, but that's just because that's my field and that's what I know. So if somebody was up here giving the same talk from the political sciences department, they would be giving different set of examples, just as impressive as some of the Creighton students that I've told you about. Creighton has done very well in federal funding over the last decade, and I focus on the last decade because since 2003, the federal funding budget, especially at NIH, has been flat in today's dollars. And if you do inflation-adjusted dollars, it's actually been reduced quite significantly. But as an institution in this 10-year period, we have actually increased our funding by about 28%. There's not that many schools across the United States that can say that. I'm not a big advocate of all of the U.S. News and World Report ratings. Of course, they are important for Creighton, for the #1 Midwest undergraduate program rating. But in ranking medical school research programs, Creighton's rank last year was 71st, and our colleagues and friends down the street was ranked 64th, and they have over 3 times the size of the faculty that we do. So our reputation nationally is very good. Now, besides being a neuropharmacologist, I'm also a card-carrying neuroscientist, and there's a lot of exciting things happening on the campus with respect to neuroscience research. Some of you may know that beginning next fall semester, we're going to be rolling out an undergraduate BS program in neuroscience. We hope to follow that up in a couple of years with a master's program in neuroscience here at Creighton also. And I tell you about neuroscience because there's neuroscience research that cuts across several of the schools at Creighton: School of Pharmacy, College of Arts and Sciences, School of Medicine, and also the School of Dentistry. Much of the research is collaborative across the schools, and much of it has federal funding. So we kind of work as a very collaborative institution. And we're coming, all of these same neuroscientists come together to have developed a very rich curriculum for the undergraduate degree program in neuroscience. So with that, I'll close. I'm going to be happy to answer questions. But again, an undergraduate research experience is one of these high-impact experiences that can not only will it be enjoyable, but has the potential to help you decide on what your career choice is going to be. I'm happy to answer questions. I would say that you don't necessarily have to wait till you decide on a major to start. If you did something in high school, in biology class or history class or computer science that you liked and enjoyed, you could look for a research experience that would allow you to continue that. Julie, you might want to weigh in on that topic. No?
Once you have a major, that might be the most common thing to do, is to find someone in that department, just because you could maybe then have a faculty member who knows you from the classroom and from the research experience as your advisor, just especially for writing letters and things for later on. But you know, sometimes you don't know what your major is until maybe junior year, senior year. So you wouldn't want to wait, you know, that long to get involved if that's something that you really are interested in. So exactly what Dr. Murray said, I would just look for opportunities and start something. I've had students who have started as freshmen in my lab, and after a year decided, I think I'm going to be a biology major, not a chemistry major, and I think I want to do research with someone else. And that's great, because I wouldn't want them to not do what they need to do or want to do to move on later on. So I think you'll find that faculty are pretty open about, you know, inviting you to come in and learn about maybe what they do even before you start, just to see if it's a good fit for you. So the question is how to find out about opportunities or get involved. So I think if you're in a class right now and you know that faculty member is working in a certain area and you're interested, definitely go talk to them because you already know them from class, so that's a good connection already. But faculty are pretty open about, you know, if you email them and say, can I come and talk to you about your research? You know, I would guess most would say yes. Some just have open door policies. You can stop by if you know when their office hours are. The thing that I wouldn't do is mass email 20 faculty. I would take a more personal approach and individually email faculty that you're interested in. So the question is there somewhere to find out about opportunities? So on the Curis website, and then I think if you have a sticker or a pen, you might have our web address. There is a student interest form that you can fill out. And so it's very, very small amount of information, your name, your net ID, and what you might be interested in. And then what happens is we have contacts in all the different departments on campus, and we'll get your information, and then we'll connect you with someone in biology or in history that will then arrange to meet with you to talk about those opportunities. And you guys have.
And then to follow up on Dr. Sukup's suggestion, which is the first step, you can then explore departmental websites and individuals' faculty websites to see, I mean, you'll know which department you want to start at. It's political science or history or biochemistry or biomedical sciences, and then look to see what kind of research is going on at faculty. If you go to like PubMed, the NIH library, and plug in Creighton, you'll see all the publications that have Creighton's derived from Creighton faculty in chronologic order. So that's another way to do it.
So will there be any time commitment in general? Because you know, for the students who work and have outside of life besides school.
Yeah. That's a really important question. It's always the first topic of conversation that we faculty have with undergraduates that come in and talk to them. And I'll give you my answer, then maybe Dr. Sukup will like to follow up. What I usually tell students is it's good to have one longer block of time in a week than several little blocks of time. So usually the kids that work in my lab come in for one 3 or 4 hour block, sometimes multiple, depending on the student and how long they've worked in the lab and what they want to accomplish, multiple 3 to 4 hour blocks. But right now I have a student that comes in once a week for 3 to 4 hours, and that allows you to prepare, to get something done, and to have continuity of your project. Do you want to weigh in on that, Julie?
Maybe I'll just add a little bit more about, you know, once my students are trained, know what they're doing, have gained a little independence, and have some ideas about, you know, what they want to try in the lab, they might be in there 20 hours a week, some weeks, but, you know, maybe 3 or 4 hours because they have 3 tests that week. So there's also some flexibility. One of my juniors right now was very busy this week, so he's going to be in the lab all day tomorrow because he didn't get to do some of the things that he wanted to do this week. So some flexibility in time in many types of projects. In projects that don't require you to go to a space to do your work, even more flexibility where you can work on your own. So.
One of the undergraduates that works in my lab gave me a 3 to 4 hour work assignment last week when I had to write 15 or 20 letters of recommendation because she's applying for a funded summer research experience at schools across the country. So, but I finished my homework assignment. I'm done. The research papers that the kids, like the examples that I showed you today, they're called refereed publications. So those papers have been submitted to a journal, and the journal had then in turn sent the papers out to 2 or 3 faculty, scientists across the country or world, asked them to review and critique the paper to see if it was acceptable for publication. And those critiques come back, and typically you have to do some modifications and then resubmit it to get it in. So a peer-reviewed publication, which is listed in PubMed, the National Library of Medicine's database, has a higher level of rigor. It's been vetted. It's been reviewed. So a paper, a high-quality paper that you might write for an undergraduate project or even as a graduate student and submit to your mentor will grade it and critique it, but it hasn't been anonymously peer-reviewed. And as an extension of my answer to your question, the reason why I tell people, including undergraduates and including people I bump into in Hy-Vee, if you want to know about a disease or a drug, not to just Google it, but go to a database that has only peer-reviewed publications. Then you'll know that the quality of the information that you're reading is a higher caliber. You can trust it. For whatever field that you're going to get into, let's say if you're going to get into a social science field, you're not expected to come in and to be an independent researcher the first week. So, I mean, the more knowledgeable you are, the better. But that just influences the slope of the learning curve. So, I think the most important thing is your interest and curiosity. If you have those two, you'll do fine, and you'll learn along the way all of the things that you need to really make a contribution to answer the questions that you develop and you're interested in.
Maybe I'll add one more thing. Just so in certain fields and in certain departments in our college, the College of Arts and Sciences, some faculty do like you to take some kind of, many have research methods courses. Maybe Augusta could comment on this too, but in sociology, I think they like you to take that course first before getting into a research project. That course could probably be taken in this sophomore year, so that means you could still, get involved pretty early on. So if I waited until all the students that wanted to do biochemical research took biochemistry, that would be the middle of junior year. So in some cases, it just doesn't make sense to wait that long. So for every different kind of individualized field, there might be a slight, difference in come in, the minute you get to campus versus need to take maybe one course or 2 courses before they feel like you're prepared to know, have that little bit of background before you start. So does that help? So the question was, if you have an idea and you want to start an independent project, how would you go about doing that? Start it on your own, talk to a faculty member?
I think is aligning with the faculty member whose interests are in that area. We have, if I have a neuropharmacology research lab, and we have some projects that we have funding to perform that research. So I have an obligation. I've told the federal government that this is what we're going to do. But we always run into things that we didn't plan on, and then students take those projects as an offshoot. So the important aspect is you have to find a faculty member, a lab or faculty member's interest that is in the right area for the project that you think is interesting. And then, of course, you'll have to convince a faculty member that it is interesting. But that's a good challenge.